like I said, today we're going to be talking about the next kind of piece of the events in the adaptive immune response, specifically in the T-cell immune response. So we saw the transport of antigens to lymphoid organs and the movement of T-cells to those lymphoid organs last time. And so today we're going to kind of get to the recognition by that naive T-cell. And we're going to see specific details of the recognition of those T-cells. And so this is sort of the general process that we're going to be talking through today, Friday, and a bit on Monday as well. So what you can see here is that our T-cell can interact with some cell presenting antigen. Whether that cell is presenting antigen on MHC class 1 or MHC class 2 will differ based on whether we're talking about a CD4 T-cell or a CD8 T-cell. And we'll have some sort of differences in terms of which cells can be that APC. But that T-cell will get some signal from this interaction, and that will activate the T-cell. And that will lead to all of the other things that are shown on this slide. This activation step that we see happening between the antigen-presenting cell and the T-cell is where we are focused today. And so today we are focused on thinking about the actions that happen downstream of the T-cell receptor. And so we are going to be looking today at the T-cell receptor and the signal transduction process that happens downstream of the T-cell receptor. When I talk about the T-cell receptor, remember that the T-cell receptor is often with a co-receptor protein CD4 or CD8, which I told you before and will tell you again later today, has a kinase associated with it in its intracellular domain, as well as these other co-receptor proteins, the CD3 proteins, that have the long cytoplasmic tails with different numbers of ITAMs, which can be phosphorylated by kinases to lead to signaling. In reality, when immunologists think about activating T cells, there are three pieces to the puzzle of activating T cells that we talk about. One is TCR signaling. There are others that are known as co-stimulatory signaling and some cytokine signaling. Immunologists often will refer to these as signal 1 and signal 2. Sometimes people even talk about the cytokine as signal 3. Today we are going to be focused on signal 1. Signal 2 and some of signal 3 come up on Friday. So today is really all going to be signal 1. If I later on make in a later day, I make reference to, well, that's a signal 1 kind of thing, I mean today. So today is signal 1. And as I get into this, I want to show you two images from a paper just to kind of give you one other bit of information about this T-cell signal 1 signaling. So officially, this is an approximation of what happens in the first 10 minutes. You can see there's a lot of stuff here, right? And this paper then goes on to say that this is what happens after that. This seems like a whole lot of things, right? So what I am going to be telling you about, while it may seem like, oh my gosh, there's so many proteins she's telling us, is actually a very small fraction of what is really going on. And every protein that I am telling you about, I have chosen to tell you about for a reason. The reason is generally because that protein is involved in some kind of targeting, some drug targeting. Or it's so important that you can't not think about it. So every protein I'm telling you about, I have a reason for. I'm not just trying to give you crazy stuff for the sake of giving you crazy stuff. when we are thinking about this signaling, we are going to have one major goal for our T cell. And so with all of the signaling I am telling you about right now, the goal is that we are going to turn on transcription of a particular gene. This gene is the IL-2 gene. It encodes the cytokine IL-2. The processes that turn on the IL-2 transcript are also all the processes that are involved in activating a T cell. And so our focus is going to be on getting to the point where we can transcribe IL-2. Pretty much any other thing that happens involving a T cell getting activated overlaps with this, so we're just going to be thinking about that. And in order to get IL-2 transcribed, you need to turn on 3 transcription factors. And so again, our goal with signaling that we see today is going to be to get these 3 transcription factors. If you only have 2 of them, you don't make IL-2. You got to get all 3. So we're going to see how we get to all 3. And those 3 transcription factors are called NFAT, AP1, and NF-kappa B. And so we are going to see the activation of NFAT, AP1, and NF-kappa B in this process today. This signaling starts with the T cell receptor that you have seen previously. So you can see that T cell receptor here. You can see the MHC and peptide are interacting with the T cell receptor. You can also see that the CD4 molecule in this case is interacting with class 2. And I told you previously that the T cell receptor has relatively low affinity for MHC plus peptide. And I said that CD4 in this case with MHC class 2 or CD8 in the case with MHC class 1 help to stabilize that interaction. In reality, there are a whole bunch of other proteins stabilizing this interaction. In reality, there are actually a whole bunch of proteins sort of outside of this area that are grabbing the cells and holding them together to sort of help out with the fact that this interaction is in fact very low affinity interaction. So there are other types of proteins that are holding these cells together while this signaling takes place. One set of those proteins are actually integrins and CAMs. So you saw integrins and CAMs last time. They had a very high affinity and are able to sort of stop cells moving in the flow of blood. They can also hold those cells together very closely so that we can actually get a long interaction between the cells so that we can get some signaling. One thing that will become important about these integrins and CAMs next week is that they also make a bit of a wall. So if there are any molecules that are secreted between the T cell and the antigen presenting cell, or between a T cell and some other cell, the integrins make a little wall so that those molecules can't diffuse away and go other places. They sort of keep things that are secreted just going between these 2 cells and don't let them go out and get lost. Immunologists have mapped many of the proteins that are on the surface of the T cell and the antigen presenting cell. So you can see, in addition to the T cell receptor and CD3 and either CD8 or CD4, which interact with class 1 and class 2, we've got some of these adhesion molecules holding together these 2 cells. We've also got some of the other signaling proteins that are involved in signal 2 in this area. And the way that this actually will work is that the T cell receptor, sort of the membrane, if you remember, we've talked about the fact that, that membrane proteins can diffuse freely in the membrane. all of the proteins that are important for this signaling will sort of get clustered together. And it's so much so, this clustering, that you can actually see it under a microscope as this thing called the immunological synapse. And so if we look, we can actually see the TCR and CD3 and all of the other signaling molecules all end up in a little cluster together on the surface of the cell, so they can all signal together and work together. The adhesion molecules make a wall around the outside to sort of hold these cells together. And so you can actually, this is actual imaging of where the adhesion molecules are, where the signaling molecule is. Here's the end-on view of the antigen-presenting cell and the T cell, and you can see them coming together. And so remember that the term synapse really just means a place where two cells are coming together and interacting. I mentioned to you before with VDJ recombination that there's this little bit of a rivalry sometimes between immunologists and neuroscientists about like whose thing is more difficult, more complicated. And sometimes I feel like this is just an example of immunologists wanting to be like, we have a synapse too. These proteins are all going to organize themselves at the immunological synapse to allow for signaling. This is also kind of interesting and important in some other ways. So you can see the immunological synapse here. You can see our TCR and MHC signaling together. You can see our integrins making the wall outside. But you can also notice this example where we have two cells coming next to each other, right? And notice that the area where the two cells are really close to each other is a little smaller than these other areas out here. There's more space outside. There's less space right where the two cells are coming together. One thing that actually also happens here is that proteins that have super big extracellular domains get shoved out of the way because there's no space for them. There's this example of this protein called CD45RA. CD45RA cannot be at the synapse, the T cell synapse, because it has this super giant extracellular domain, and it just won't fit in between the two cells. So it physically is pushed out of the way. It physically gets removed from the synapse. The reason why that's important and cool is that CD45RA actually has phosphatase activity. It can remove phosphates from stuff. And a lot of the signaling we're going to see is adding phosphates to stuff. CD45RA could turn off our signaling. So we actually just take this off protein and shove it far away. It can't fit in this area, so we can't have it accidentally turning off this signaling. And so the organization of these proteins between the T cell and the antigen-presenting cell is particularly interesting. Once the T cell gets in contact with the antigen-presenting cell, the first step of signaling will occur. This signaling is really similar to, I've basically shown you this exact part of the signaling before. We're just going to go further with it later on. This is officially known as the receptor proximal part of T cell receptor signaling. That means it's the part that's close to the receptor. We're going to get further down the pathway, further away from the receptor. Right now we're at the top, so we're calling this receptor proximal. So originally, before MHC plus peptide came around, the T cell receptor was diffusing in the membrane with CD3 in some way. And CD8 or CD4, whichever one is in our diagram on this day, is diffusing in the membrane. And they're not near each other. That's what this is supposed to show us, is them being not near each other. But if it's supposed to be on a slide, it's kind of hard to make them not near each other and still both be on the same slide. This is also supposed to show them not near each other. Our CD4 or CD8 molecule has a kinase that travels along with it. That is this intracellular kinase. It's officially a different protein, but it associates very closely with CD4 or CD8. This kinase is called LCK. Officially, LCK is the kinase in this case that works with CD4. The one that works with CD8 is part of the same family. If you call them both LCK, I do not care. And when MHC plus peptide comes around, it will bind to the T cell receptor, and it will also bind to CD4 or CD8, which bind to kind of the back part of the MHC molecules. And that will make our CD4 get close to the T cell receptor. So this is supposed to show them far from each other in the middle. This is supposed to show them close, or closer. And when they're close, now our kinase, LCK, can phosphorylate the ITAMs that are on the CD3 molecules. And so you can see the phosphorylations as pink dots that are added to those ITAMs because our kinase, LCK, has been brought into proximity to CD3. This is, in fact, an example of something called the induced proximity model of signaling. Yep, Jamie.
I feel like I might have just missed something.
Okay.
So I think what you said is so when MHC plus peptide binds to the T cell receptor,
Mm-hmm.
does it also bind to CD4 at that point?
Yeah, so CD4 binds to the back part of MHC.
So how does is it close enough that it can still bind, or is it moved?
It will actually, at some point CD4 will diffuse by, and it will sort of catch, and they will all be brought together in close proximity.
It's not like it's pulled, it's just...
Yeah, I mean, you can kind of think about whether it's pulled, the order of events. It could be slightly pulled.
Okay.
It could potentially engage first, but that's a really hard thing to actually study. The moral of the story is CD4, TCR, CD3 all get pushed together. The kinase, LCK, that is traveling along with CD4 or CD8 now phosphorylates the ITAMs that are on CD3. So we get the pink dots. Previously, we had some ITAMs, which are here as like the yellow rectangles, that didn't have anything on them. Now, we have ITAMs with phosphates on them. At one point during my time in college, I was talking to a friend of mine who is not a science person, and I was trying to explain some parts of immunology to her. And I told her that it was all about shapes and whether or not shapes match or not. And she was like, oh, so it's basically Sesame Street. I was like, yes, exactly. And so we're going to think about this very much in terms of shapes and matching. So now we have a new shape. It also has a new charge because of the phosphate addition. But the shape is different. And as a result, something that previously could not bind in this region now can, because there's a shape available for it to bind. The thing that is going to come and bind is a protein known as ZAP70. So ZAP70 has this domain that specifically binds to phosphorylated ITAMs. So before, ZAP70 wouldn't come hang out in this area. Once we have the phosphorylated ITAMs, you can see ZAP70 is pulled into this location. Again, it's hard for them to really draw things with ZAP70 being far away, with it still being on the slide. So now you can see we bring ZAP70 into this signaling process where we didn't have it before. ZAP70 has is an enzyme, and it has a specific enzymatic function. ZAP70's enzymatic function is that it is a kinase. So ZAP70 can do more phosphorylation when it comes by. We don't need to worry about exactly who or what ZAP70 is phosphorylating. We'll just say ZAP70 starts phosphorylating stuff. In reality, this showed that LCK got all of the ITAMs. Really, LCK does some and ZAP70 does some. Don't stress about that. You can just kind of say ZAP70 comes in. Okay? You can also see ZAP70 itself gets phosphorylated. So, Now we get to play with shapes again. We now have a phosphorylated ZAP70, some different phosphorylated stuff here. It's a new shape that can allow new things to bind. So new proteins can get recruited to this area that couldn't get recruited here before because their shape that they were looking for was missing. But now their shape is here, so they can bind. Also with phosphates, there's also a charge involved. And so now we are going to bring in the next step of this pathway. There are actually two proteins that act at this step. I'm going to tell you both of them, and I'm going to tell you they're basically interchangeable. So I'm going to mention them both just because, for correctness' sake, I guess. But if you can think of kind of both of them as doing the same thing. So these two proteins are known as LAT and SLP76. This is a slightly off-topic thing, only slightly, but I'll just tell you, if I was doing this for B cells, it'd be a different kinase of this same family, a different kinase of this same family, and then there'd be one called LAB instead of LAT, and then there's one called SLP67 instead of SLP76. So like it's the same thing in B cells. LAT in fact stands for the linker for activated T cells. So LAT and SLP76 are both the same type of protein. They both play the same function in this process. You can sort of get that one word for what they do is linker, which is what the L stands for here. Although I usually use a term called adaptor proteins. So these are two adaptor proteins. So now we're bringing in the adapters. So here you can just see, just figure from your textbook, we've got the phosphorylations from ZAP. Now we're bringing in LAT and SLP76. And the bottom is also showing us some information about adaptor proteins. This figure is calling them scaffolds. All the same thing. So in non-COVID times, this makes me feel sad. In non-COVID times, I used to go to South Africa every year. And when I went to South Africa, one thing I would have to pack in my suitcase was an adapter. What was the adapter I would pack? What was the point of that adapter? Yep, Sebastian. Oh. Okay, Abby, what were you going to say? Yeah, so basically, the plugs there are one shape. The plugs here are another shape, so I want to charge my phone. Does the adapter actually do anything to the electricity? No, it literally just changes the shape from one form to another, right? It lets us sort of change which things can talk to each other, but it has no other function whatsoever. That's what these adapter proteins are. These adapter proteins, basically, particularly when they get phosphorylated, which they might get phosphorylated by ZAP70, are basically proteins that bind to a whole bunch of stuff, and they don't do anything. They just serve as a way that those proteins can talk to each other. So here you can see we have this adapter. It's green. It has some pink dots on it. And as a result, all sorts of other proteins can come in and bind and dock and suddenly be next to each other, whereas previously they were all over the cell. So the adapter is just basically a place where a whole bunch of other proteins, you can also see this at the top with LAT, with SLP76, it's just a place where a whole bunch of other proteins can bind and start talking to each other that normally would have been different things like South Africa plugs and US plugs. Now we make South Africa plugs and US plugs talk to each other because we have something that can fit in between them and pull them together. That's my adapter, and that's what the adapter protein does. It's basically just a protein with a whole bunch of binding sites that lets a whole bunch of proteins bind to it, come together, be in proximity, and start to signal together, but it itself has no function and no enzymatic activity. So now we get these adapter proteins recruited. And once we have our adapter proteins recruited, as you can kind of see on the, really both the bottom and the top, I was going to show you the bottom, we bring in lots of other proteins. And lots of things can start happening. And so this is where this pathway starts to branch into all those crazy branches I showed you before. And so what we're going to do now is we're going to follow one of these branches. So we're going to follow one protein that comes in and binds LAT and SLP76, and we're going to go from it all the way to a transcription factor. Then we're going to come back to LAT and SLP76 and do a different branch. So we're going to sort of see how a bunch of things branch at LAT and SLP76. So the first pathway that happens with LAT and SLP76 is known as the Ras-MAPK pathway. So I'm going to draw some awesome things on the board to help us understand the Ras-MAPK pathway. So we can imagine that we have a cell, and it has a protein. And that protein helps the cell do cell division, which just to make everyone's life easier, we'll remember includes mitosis, as well as doing some transcription. I'm going to make up a name for this protein. This is not actually the real name for this protein, but our lives are going to be easier if you think about this made-up name, okay? So this is referred to often as MAP, or a mitogen-associated protein. Mitogen is a thing that generates mitosis. So this is involved in mitosis. So we got this protein, we'll call it the MAP, the mitogen-associated protein, okay? There is not actually a protein called MAP, but again, our lives are easier if we just pretend there is for right now. So there are some proteins that serve this function. They act as MAPs. Those proteins get turned on by getting phosphorylated. If you were in charge of the world, what would you call a protein that turned on this MAP? Remember I told you that it gets turned on by being phosphorylated. Yes, Jamie? PMAP. Okay, so this is going to be PMAP. What makes PMAP? Oh. Yeah, Hannah. It would be a kinase, probably like MAPK. It would be called MAP kinase, right? It's a kinase that phosphorylates MAP. So MAP kinase phosphorylates the MAP, right? Yeah? Make sense? Okay. MAP kinases, which are a real class of proteins, MAP kinases get turned on by phosphorylation. If you were in charge of the world, what would you call a protein that turns on a MAP kinase? Yes, Jamie? You would call it a MAP kinase. MAPKs exist. They're a class of proteins. Okay. So MAP kinases get turned on by phosphorylation. If you were in charge of the world, what would you call a protein that turns on a MAP kinase? Yeah, Rishi? You would call it a MAP kinase. MAPKKs, MAP kinase, are a class of proteins that actually exist. They also have to get activated. They have to get turned on. They get turned on by a protein known as Ras. The name of this pathway is the Ras-MAPK pathway. hopefully, you can see how it got that name and understand how this general pathway works. If you look at this image from your textbook, one of the things that is turned on by LAT and SLP76 is RAS, which will then start this whole cascade. So RAS is the first thing that can be turned on downstream of our adapters, and that will turn on this MAP kinase pathway. In reality, there are many different proteins in the cell that can serve the function of MAPKKK. A bunch of MAPKKs, there are a bunch of MAPKs. So we often see specific versions of these in this signaling cascade. So very frequently, the name of the MAPKKK that we care about is RAF. How do they write this? Okay, good. The name of the MAPKK we care about is MEK. The name of the MAPK we care about is ERK. And the name of the MAP we care about is FOS. I don't know why they picked those names. Those are just the names. And so, in fact, if you look, you can see RAS getting turned on here, then RAF, then MEK, then ERK, and we turn on FOS. There are other versions of this Ras-MAPK pathway. One parallel version of it involves a different MAPK. That MAPK is known as junk. Junk phosphorylates a protein known as Jun. And Jun actually, junk is actually Jun kinase. That's what junk stands for. But this is just another parallel version of the same MAP kinase pathway. So you can see this is the entire Ras-MAP kinase pathway that we care about here. When FOS and Jun are both made together, when their powers combine, they don't make Captain Planet. Instead, they are called AP1. And so FOS and Jun together are called AP1. And so the Ras-MAP kinase pathway, which you can see on this slide, gives us the first of the 3 transcription factors I told you about. Remember I told you that to make IL-2, we needed to get 3 transcription factors. Here's one, which is AP1. It comes from the Ras-MAPK pathway, which you can see here. Oh, look, Sydney answered about MAP kinase 2. I could only see it when I walk over here, though. Okay, so this is one thing that happens downstream of LAT and SLP76. And it gets us one of our transcription factors. We're now going to look at another thing that happens downstream of LAT and SLP76. So here you can see we're going back up to LAT and SLP76. So LAT and SLP76, one thing they do is they signal to RAS. And RAS is actually on this slide. I just covered it with a white box to get rid of confusion. But so RAS, the RAS pathway is like doing its thing over here. While the RAS pathway is doing its thing, we get this other branch doing its thing. And this other branch starts with a protein called PLC gamma. In order to make some sense of PLC gamma, I want to tell you what PLC stands for. So PLC stands for Phospholipase C. If we look at the name Phospholipase C, This will tell us some things about PLC gamma and will help us understand what PLC gamma does. So first, let's look at the suffix of phospholipase C. What's our suffix? Lipase or just? ACE. So that tells us what? What? It's an enzyme. It has an enzymatic activity. Okay? Then we've got, before the ACE, we've got phospholipe, so phospholipase. So what does it mean that it's phospholipase? Rishi. Okay, something about phospholipids. Krishna.
Well, break up phosphate.
Break up phosphate or break up, what about the lipase part? Yeah, Jamie.
So is it breaking up phosphorylated lipids?
It's breaking up phospholipids, yes. Yes? I told you that's what we were doing today.
It's my face. Then how do you know how they're looking at you? It's just looking.
Are you enjoying this?
Yeah. We're going to have cyclosporin in a little bit.
Okay.
I'll do anything to avoid grading.
Uh-huh. Okay, you want to come learn about cyclosporin?
You guys got it.
All right, so phospholipase. Can you remind me about phospholipase, what we were saying phospholipase is? Yeah, Karen? It cuts phospholipids. So specifically, phospholipase cuts phospholipids. It actually cuts a few phospholipids, but we only care about one thing that it does. Phospholipase C, particularly PLC gamma, will take this phospholipid that you see here. It's known as PI45 bisphosphate, or known to those of us in the biz as PIP2. PLC gamma will take PIP2 and will cut it into these two products. One is called inositol 1,4,5 triphosphate, also known as IP3. The other is known as 1,2-diacylglycerol, or DAG. And so PLC cuts PIP2 into IP3 and DAG. One other thing you can notice is that PIP2 has some nice long phospholipid tails that are in the membrane. DAG sort of stays up here at the membrane because it has the tails. IP3 can sort of diffuse around this. All right, so we have the tails. They're in the membrane. We're going to have DAG give us one transcription factor, IP3 give us the other transcription factor. So first we're going to watch what happens with IP3, then we're going to come back to DAG. I will tell you, the IP3 part of this is the one that every immunologist knows really well. It's the really famous one in immunology. And for a long time, I always felt bad about DAG, like it was getting left out. So that's why I have to make sure I tell you some things about DAG. All right, so IP3 will be made with the cleavage of PIP2, and IP3 will lead to calcium release from the ER. Normally, your cell keeps all of the cytoplasm, or all of this, that works, actually, that grammar works, keeps all of the cytoplasm free of calcium. So no calcium in the cytoplasm. All of the calcium gets hidden in other places, like the ER. The cytoplasm usually has no calcium. IP3 makes the ER release all its calcium. So the calcium gets released from the ER because of IP3. So IP3 binds to the IP3 receptor, which is sitting in the ER membrane. So you can see the IP3 receptor here, which is in yellow. It's binding to the ER membrane. IP3 binds, and as a result, calcium leaves the ER. So calcium is the blue dots. There was previously no calcium in the cytoplasm, only calcium in the ER or outside of the cell. When IP3 bound to the IP3 receptor, that opened a calcium channel, so suddenly all of this calcium flooded into the cytoplasm, just because it was going down its concentration gradient from high to low. So now we have a cytoplasm full of calcium. That calcium will bind to calcium binding proteins. I know, shocking. That calcium will bind to calcium binding proteins. The calcium binding protein is called Calmodulin, which also turns on a target. So we're officially going to have Calmodulin, and then Calmodulin, so here you can see it's like a wrench. It was folded up, now it becomes wrench-like when it binds calcium. Calmodulin turns on calcineurin, so we're going to get Calmodulin and then calcineurin. And now we're going to get the most important part of this process. At the beginning of this process, before we had signaling, our transcription factor NFAT had a whole bunch of phosphates on it. And because our transcription factor NFAT had a whole bunch of phosphates on it, it was not allowed in the nucleus. That actually blocked it from going to the nucleus. When calcineurin is active, it removes all the phosphates from NFAT. And now NFAT can go into the nucleus and begin transcription. And so now we have actually turned on our second transcription factor, which is NFAT. So the calcium release leads to Calmodulin and then calcineurin activation. That leads to the dephosphorylation of NFAT. NFAT goes to the nucleus, does transcription. As I mentioned to you before, this is the kind of most famous part of this pathway to most immunologists. And the reason why this is the most famous pathway to most immunologists, or part of this pathway to most immunologists, is because we have a drug that blocks what is shown on this slide. And so we have a drug that will actually make it so that this part of the signaling doesn't happen, so that NFAT just sits there with its phosphates on it, never goes into the nucleus. That actually blocks T-cell activation. It's great for patients who are getting transplants, who don't want to reject their transplants. This drug is actually why we can do transplantation. Transplantation kind of wasn't really a thing before this drug. It also can be used to treat a whole lot of autoimmune disorders that involve T cells. The bad news is it kind of stops all your T cells. So you also can't really deal with a lot of infections at the same time. But if you need a kidney, it can be really important for you. So this drug is called Cyclosporin A. And Cyclosporin A is shown here as like the orangey pill-shaped thing. Cyclosporin A gets in between Calmodulin and calcineurin. So we've got the calcium coming in, you've got Calmodulin binding it, being ready to go. But Calmodulin cannot talk to calcineurin, cannot activate calcineurin. And as a result, NFAT just hangs out inactive forever. Can never go to the nucleus, can never give us transcription. And so we can never get IL-2 or any of the other actions that turn on T cells. And so this drug is sort of, like I said, pretty revolutionary in the immune system. So now we've seen 2 of our 3 transcription factors get turned on. Our final transcription factor is NF-kappa B. Yes, it is the same NF-kappa B that's important in innate immune responses. It's also important in adaptive immune responses. It's actually important in pretty much all immune responses. It was originally described as something that was involved in transcribing the kappa chain in B cells. the nuclear factor for the kappa chain in B cells. While NFAT was the nuclear factor for activated T cells. But now we know they all do both. NFAT is turned on downstream of DAG. I didn't want NFAT to, NF-kappa is turned on downstream of DAG. Didn't want DAG to get left out. So DAG is also one of those products of PEP2. It leads to a bunch of signaling, including this protein kinase called protein kinase C delta, and that turns on NF-kappa B, our third major transcription factor. And so now, here is the entire figure that your textbook gives you. You can see that we have our TCR and CD3 molecule interacting with our CD4. This brings in LCK to do some phosphorylation. This gives us some new binding sites, including a binding site for ZAP70, which is able to phosphorylate more things. This gives us binding sites for LAT and SLP76, which also get phosphorylated. Once LAT and SLP76 are phosphorylated, we turn on RAS and get the Ras-MAPK pathway to get AP1. We also have PLC, which splits PIP2 into IP3 and DAG. The IP3 pathway gives us NFAT. The DAG pathway gives us NF-kappa B. And now we happily have all three of our transcription factors together, and we can do our goal of turning on IL-2. So that's a big piece of how we think about activating T cells. Everything I have told you here involves the details of what I told you before is called signal 1. So remember that we do also have some other things going on here, signal 2 and signal 3, which I am going to spend some more time talking about. Signal 2 is like crazy important in immunology. And a protein that's involved in signal 2 is called B7. Is one part of activating T cells, which is signal 1. We do also have to talk about signal 2 and signal 3. But I want to tell you a couple of things that you need to know before thinking about signal 2. And the thing that I want that you need to know when thinking about signal 2 has to do with dendritic cells. I've sort of hand-waved about dendritic cells before. Dendritic cells are innate immune cells, but they're usually thought of as kind of forming the bridge between the innate and adaptive immune system. We've talked about antigens being transported to the lymph node via the lymph. But we can also have antigens transported to the lymph node via dendritic cells. Dendritic cells come in different forms. Particularly the forms that we care about today are called immature or mature dendritic cells. Immature dendritic cells really kind of look pretty much like monocytes. And those immature dendritic cells are really, really, really good at doing phagocytosis. They love to do phagocytosis. They'll eat up anything that's around. However, when they actually get a stimulus from a PRR, they mature. And DC maturation leads to lots of changes. Some of those are changes in structure. Some of those are changes in how much the cell likes to do phagocytosis. So the cell kind of stops doing phagocytosis at that point. It's like, I already caught something, I don't need to catch anything else. And that cell also starts to move. So that cell is now going to move to the lymph node. And it's also going to put a whole lot of additional proteins on its surface. So that mature dendritic cell is going to have a ton of proteins on its surface. And so those activated DCs, you can kind of see happening here as well, that we originally have some immature dendritic cells in tissues, they will start doing phagocytosis. If they phagocytose something, they're going to head to the lymph node. And they're in fact going to really head to the T cell area of the lymph node. And they are going to also, in their process of maturing, start making tons of proteins to have all over their surface. As a result, dendritic cells are by far the best cells for activating T cells because they have so many proteins on their surface that can do things like give that signal to or other signals to help out the T cell receptor signaling. And so dendritic cells are the all-stars at turning on T cells. Really, you don't have a dendritic cell involved, your T cell's not getting turned on well, not getting turned on correctly. That actually leads to a little bit of an issue that I skipped over before related to antigen presentation. And so I'm going to start with that antigen presentation related issue next time and then move into thinking about signal 2. And that's because dendritic cells are the best at doing signal 2.